Welcome to Let's Learn Plants. Today, we're taking a really fascinating look under the hood, literally into the microscopic machinery that drives plant life. We're going to break down exactly how plants manage to move essential nutrients, ions, and water exactly where they need to go. And they're often fighting right against the laws of physics to do it. It's a pretty incredible system.
Okay, let's dive into this. Here's our roadmap for this explainer. First up, the plant cell gatekeeper. Second, forces driving solute movement. Third, the Nernst equation test. Fourth, the transport machinery. Fifth, carriers and cotransport. And finally, kinetics and power. Section 1, the plant cell gatekeeper. So every living plant cell has this boundary, right? The plasma membrane. It's a protein-lipid bilayer, but it is definitely not just a passive wall. It's an incredibly selective gatekeeper. By strictly regulating exactly what goes in and out, it maintains the cell's internal stability and that crucial turgor pressure. That's what literally keeps the plant standing upright. Without this strict barrier, the cell just couldn't take up nutrients or flush out waste.
Moving on to Section 2, forces driving solute movement.
Now that we've got the boundary figured out, how do things actually cross it? Well, there are basically two main ways: passive and active transport. Passive transport is your spontaneous, downhill movement. It strictly follows Fick's first law of diffusion, meaning molecules just naturally flow from higher free energy down to lower free energy until they hit equilibrium. There's no energy spent by the cell at all. But active transport, that's the heavy lifting. This is pushing molecules uphill against their gradient. So obviously it's not spontaneous. The cell actually has to burn direct energy, usually by hydrolyzing ATP, to make this happen. To really get why a molecule moves passively or needs that active shove, we've got to talk about chemical potential. Think of this as the escaping tendency of a substance. Molecules just naturally want to escape from an area of higher chemical potential to a lower one. And there are a few major drivers for this escaping tendency: concentration, temperature, pressure, and, this is absolutely crucial for charged ions, electrical potential. When we're dealing with ions, we actually call the whole thing the electrochemical potential. And we can actually calculate this force. Think of this formula as the secret math dictating the cell's behavior. Mu, right there, represents the chemical potential. It equals mu naught, which is the standard chemical potential under reference conditions, plus the gas constant R, times the absolute temperature T, times the natural log of alpha, which is the activity or basically the effective concentration of our solute. So at the end of the day, the driving force for passive transport is literally just the difference in this chemical potential between the inside and the outside of the cell.
Section 3, the Nernst equation test.
So let's say we're looking at a specific ion inside a plant cell. How do we actually know if it just drifted in passively or if the cell had to burn energy to pump it in? It gets tricky because membranes have unequal permeability, right? So a slight separation of charges happens naturally, creating a diffusion potential across the membrane. Basically, we need a way to untangle the electrical forces from the concentration forces. Enter the Nernst equation. This is our ultimate scientific detective tool. It lets us pinpoint the exact electrical voltage where an ion's electrical driving force perfectly balances out its concentration driving force, meaning zero net movement. In a typical biological setting, that voltage difference equals 59.2 millivolts divided by Z, the charge of the ion, times the log base 10 of the outside concentration over the inside concentration. So here is how you'd actually run this test. Step 1, you just measure the actual internal concentration of an ion. Step 2, you plug the numbers into the Nernst equation to predict what that internal concentration should be if it were just passively diffusing based on the cell's voltage. Then, step 3, you compare them. If your measured concentration matches the Nernst prediction, boom, it's passive transport. But if that measured concentration is way off from the prediction, well, our detective tool just caught the cell in the act. It means active transport is happening. The cell is definitely spending energy there. All right, Section 4, the transport machinery. Moving from the math of the membrane to the actual physical proteins doing the work, we've got three main categories: channels, carriers, and pumps. Channels are basically selective pores. They are always passive and they are lightning fast. We're talking millions of ions zipping through per second. They're often controlled by special gates that pop open or shut based on voltage or chemical signals. Carriers, on the other hand, are a lot slower. They actually have to bind a specific substance, physically change their shape to carry it across the membrane, and then release it. And finally, pumps. These guys handle primary active transport. They're slow but incredibly powerful, using direct energy like ATP to forcefully shove substances uphill against their gradient.
Section 5. Carriers and co-transport. Let's zoom in on those carrier proteins, because they fall into 3 really cool types. First up is the uniporter, which handles passive facilitated diffusion. Honestly, think of it like a simple revolving door. It just moves a single substance down its concentration gradient. Next, we get into the secondary active transporters. The symporter is kind of like a subway turnstile, where 2 totally different substances have to enter together, and they both move in the exact same direction. Then there's the antiporter. This one acts exactly like a seesaw. As one substance moves down its gradient into the cell, the kinetic energy of that movement physically pushes a completely different substance up and out of the cell in the opposite direction.
Which brings us to our final stop, Section 6, kinetics and power. Because carrier transport relies on grabbing onto an active site, we can actually study it using enzyme kinetics. The first big parameter here is Vmax, or the maximum transport rate. This is what happens when literally every single transporter binding site is occupied. It's totally maxed out. And because it measures this absolute maximum capacity, Vmax effectively tells us the total number of those specific transport proteins currently sitting in the cell membrane.
The second key parameter is Km. This is the solute concentration exactly at half of the Vmax. Km is super useful because it's a measure of affinity. It helps us understand the nature of those binding sites. Basically, a lower Km value means the transporter has a really high affinity for its target. It can easily snatch it up even if there's barely any of that solute floating around.
And this brilliantly illustrates the big question: what actually powers all of this machinery? Well, in plant cells, the master engine, the primary electrogenic pump, is the H ATPase. Using ATP, it is constantly, actively pumping protons, or H ions, right out of the cell. By continuously shoving those protons outside, the H ATPase builds up a massive electrochemical gradient across the membrane. This is known as the Proton Motive Force, or PMF. You can think of it as the ultimate cellular battery. It's just stored free energy waiting to be used. Those protons desperately want to flow back inside, down their gradient. And when they finally do, they rush back through those secondary active turnstiles and seesaws we talked about. That rush provides the sheer power needed to drag other essential nutrients in or to pump toxic stuff out. It's just this beautifully elegant cycle of generating energy and putting it straight to work. So we've seen how plants use these highly regulated gatekeepers, how they balance some pretty complex electrochemical forces, and how they run these sophisticated molecular turnstiles just to move tiny molecules around. But it leaves us with a pretty massive question. If plant life relies so heavily on this beautifully delicate, perfectly balanced proton gradient just to survive, how do sudden environmental stresses, things like a sudden freeze, a drought, or really salty soil, how do they disrupt this microscopic battery? It's definitely something to chew on the next time you're just looking at a simple green leaf. Thank you so much for joining me on this explainer. Keep learning and stay curious.